off we go. Okay, welcome everybody. I'll be talking today about work that I'm doing as part of my master's research. It basically looks at the effect of restoration on community structure, both the fish and benthic invertebrate community structure in a northern Nova Scotia stream. This work was done in conjunction with St. Francis Xavier University under the supervision of Dr. Barry Taylor, Charles McGuinness at the Department of Fisheries and Oceans, and a local nonprofit organization, Habitat Unlimited. So just before I get into the actual specifics of the talk, let's try that there. I just want to give you a little bit of a background about why we'd be interested in river restoration at all. So just from a perspective of Nova Scotia alone, we know that river restoration really got kicked off in Nova Scotia with work done by Bob Rutherford and Charles McGuinness in the late 80s, early 90s. And this has really developed up into out of that has developed this Nova Scotia Salmon Association, Nova Scotia Liquor Corporation Adopt a Stream program. It's really come out of that work. And to date, that program has funded over 40 different groups, and you can see some of the stats there, over 75,000 trees. And they're restoring about 200,000 square meters a year. So through that program. And in addition to that program, there are many other programs out there as well. So the Atlantic Salmon Conservation Foundation funds projects in Nova Scotia, as does Eco Action, Sage Environmental, and several others. So really what I'm trying to impress upon you is that there is currently lots of restoration work being done on that. And it's almost entirely based on that original work done by Bob Rutherford and Charles McGuinness. One of the groups that's doing on it, as I've already alluded to, is a local nonprofit group here in Antigonish, Habitat Unlimited. Now this group is been around for about 20 years now. It was actually founded and actually is continuing the work that was initially started by Bob and Charlie. That work is not only just the straight restoration work, but also sort of the research and the investigation side as well, which makes Habitat Unlimited a bit of a different nonprofit organization in that they do partner with Sanivex and do a fair bit of research. All of this is sort of becoming fresh because not only are we doing a lot of it, but with recent changes to the salmon stocks on the eastern seaboard, restoration is really being looked at as a possible way forward. So just to give a very brief summary of what I'm alluding to here is that the eastern seaboard, of course, has experienced recent declines across the board in its salmon stocks. Some areas have obviously been harder hit than others. The Gulf of Nova Scotia is doing relatively okay, but other regions such as the southern uplands or the inner Bay of Fundy, the species are listed or are becoming listed as endangered. So with that, people have been looking at the causes, which is generally accepted as at-sea mortality, but where this leads back to restoration is that people are thinking the way forward is to try and maximize in-stream production, and so restoration is being looked at. So really what I'm trying to prove or just demonstrate here is that there's lots of restoration being work done, and it looks like there's going to be even more done in the future. So it is very important, therefore, for us to understand the principle behind restoration and exactly what's happening in terms of the restoration and what the exact effects are. Okay, so that's really all I'm trying to go with that there. So if we look at the literature to see what has been done in this field, we see generally most of the studies are quite positive towards restoration. So we have the initial work that was done by Charles McGuinness and Bob Rutherford in 1990. It was published in 1994, looking at basically showing positive effects, increases in rearing areas for juveniles. And there's been several others through the years since. So generally quite positive along the board, and that's just, of course, a subset of what's been out there. But of course, you never have anything that's perfectly positive. So through the years, there's been a few papers that, although not really condemning restoration, not suggesting that river restoration shouldn't be done, they have been calling into question some of the claims made by restoration or questioning the mechanism by which we see restoration being done. So I've just sort of once again put a little subset up there, but we see Riley and Falk. They were concerned that some of the increases in community and usage was possibly due to immigration Roney and Quinn worried whether or not there might be some movement impairment, especially to other species. So every once in a while I'm getting a beep there. I'm not sure why I'm getting that, but anyway, okay. Studying it all, this was a very interesting one. They actually did a meta-analysis of a bunch of different restoration programs, and they found that, or they raised concerns that a lot of groups out there doing restoration really weren't connecting with the underlying ecological processes, that they were just that they were doing the restoration work, but didn't necessarily connect it back to limiting factors and actually trying to improve the underlying ecological process and thus the ecosystem. And then very recently, Langford et al. In 2012, they did some work in Scotland, and they're actually wondering about, or sorry, in England, and they were wondering about, they were seeming to find impacts to a lot of other species, so non-salmonids, and that didn't seem to benefit from various forms of restoration. The other sort of one is actually coming out of my own lab, in that one of the things that we're finding, curiously enough, is that the invertebrate community, the benthic invertebrates, are not responding to this river restoration. We would expect that they should, because increasing the stability of the stream, increasing the health of the river, should, as a result, increase the benthic invertebrate community should respond to that. And we didn't really find, they're not really finding a response. Now, this is a bit of in contrast to what Harris found in '95. He found actually positive effects to the invertebrate community. So certainly there's some questions there anyway. So, and as I say, these aren't really negative on it, but just calling into question exactly how we do it, and whether or not we're doing restoration in the right way, or do we fully understand what's happening when we do it. If we look at the studies, we can sort of break them down into three general categories. Most of the studies out there are actually how-to guides, or what I call technique validations, where somebody is attempting some new type of restoration, and they just trying to validate that that the community the fish community, typically the salmonic community, has responded positively to that. So that's generally most of them. They're not actually exploring underlying scientific principles. The sort of second category that we see in previous studies is hydrology. So there's a lot of that sort of take on more very earth sciences or geological sort of basis. These are connected with sediment transport, but mostly with flood references. So control of flooding, the effects that are caused by flooding and stuff like that. So that's sort of another sort of major category in the literature. And then the third category that we see is biological in nature. So these are ecology. There's a heavy salmon and trout emphasis, so and sport fish emphasis, I guess I could say, on these sort of works. There's been some work done, but really not a lot on invertebrates, and little to no work on what I would call the bait fishes. So essentially your non-sport fish, your minnows, your dace, your sticklebacks, your chubs, etc. So so that's sort of a very brief overview of where we are at with what we know and we don't know in terms of literature, or where we're focusing our literature. So if we look at that literature, we can definitely find a few knowledge gap areas. Very often we don't know what is currently limiting our streams, either on a local basis or on a sort of a regional level basis. So the current limitations, is it still physical habitat? Should we still be applying physical habitat restoration? Or have we remediated that, and we have some other limitation that's bigger? So current limitations is certainly one area that's lacking a little bit in the literature. The specific effects of restoration, what exactly are we causing? There's been some work done on this, sediment proportions and stuff like that, but there's still a lot to be answered there in terms of what's exactly happening. What's going on with the invertebrates in restoration? Basically, we're finding mixed results, some positive, some that saying the invertebrates are not responding. So the question is what's the connection there? What's going on? Why is it responding here and not there? Are we truly seeing stream bed stabilization? Are the materials moving at a rate that's concomitant with a natural system, or are they moving and still at an accelerated or decelerated rate? So are we the underlying sort of movement or hydrology of materials, but the underlying movement of the stream bed, is that is that decreasing, stabilizing, coming more into the equilibrium that we would expect in a natural healthy system. And lastly, is there a connection between basically the geological and the biological? So there's not been a lot of work done in that area, and this is something that is, if we want to get at understanding from an ecosystem management perspective, when we're trying to promote certain ecological processes, that certainly needs to be firmed up. So we have lots of questions. Any of you that worked in science know that you always have lots of questions. It's about finding opportunities to answer those. So we had those questions in 2009, 2008, 2009, and when the opportunity came along. The opportunity in this case was the highway bypass around Antigonish. So in the map that you see up here, this was just taken from one of the planning sessions. This is for the new twin highway coming around Antigonish, so that's in purple. Okay, the existing Highway 104 is here. I don't know if you can follow my mouse, but it's along right there. The town of Antigonish is right here. The Gulf of Nova Scotia is due north. Cape Breton would be off here to the right side of the screen. So this is an opportunity because when they built this highway, as they're building this highway, they had to. It was unavoidable that they would have to destroy some fish habitat. You can see the blue lines here of all the streams and wetlands that were impacted by this infrastructure expansion, and so under at least the old terms of the Fisheries Act, the habitat destruction had to be compensated, and so Nova Scotia Transportation Infrastructure Renewal was compensating. They were going to pay Habitat Unlimited to do some restoration work, but they required a monitoring plan. So that's what actually that work and the monitoring plan was what formed the backbone of the study and allowed for me to be hired. So based on this, we decided to try and refine it down to a few sort of more specific research questions. One for this stream here that we're studying, and I'll show you the exact stream here in a second. Is physical habitat limiting? Is that still a concern, or has our 20 years of work really alleviated that issue? We want to know, is the fish community energy limited? So if they're not physical habitat limited, is there an energy limitation, something having to connection with the benthic invertebrate community? And lastly, we wanted to know what's happening with the benthic invertebrates, because as I said, the previous work that we're sort of working on now seems to suggest that they're not responding. So are they limited by physical habitat, or is there some other factor at play here? Is it substrate stability, the ability of the system to retain leaf litter and thus energy for the benthic invertebrates, or is there a litter colonization issue? So those were sort of our research questions. Based on that, came up with a series of hypotheses. Sort of the main four that I'll allude to or discuss in this presentation are here. Restoration basically should increase the amount of available habitat. So as we increase the habitat, as we restore the system, we would expect, using our standard measures, to see increases in habitat for some wanteds. As I said, using standard measures. We would expect that if we restore these new reaches, that we would see local increases in fish populations. So the fish community should respond positively, is our hypothesis, and that we would see increased numbers in fish in and around these areas that we restored. Restoration will decrease the rate of sediment transport, and we should see an increase in leaf litter. So if we are working to stabilize the movement of the stream back to more of a normal equilibrium of erosion deposition, then we would expect that the sediment transport, or we're guessing that the sediment transport rate will decline, and that leaf litter should increase, because it won't be removed from the system, won't be chewed up as fast by the turning over of the substrates. And lastly, the hypothesis is that the invertebrate biodiversity should increase after restoration as well. So here's the specific study site. This is, as I alluded to before, it's done just outside of the town of Antigonish here in northern Nova Scotia. The stream of interest is Brierley Brook, the same system that Bob Rutherford and Charles McGuinness did the original work in. So we can see here, this runs along here. This is the existing Highway 104. The town of Antigonish is just up here in the upper right-hand corner. There's a wetland right here that I've highlighted, and it's right through along the edge of this wetland. That's where the new highway was put. So the new divided highway is right there. So this was showed overwintering habitat that was lost, and then there was, of course, erosion and sedimentation issues affecting the streams down, affecting Riley Brook as it drains into Riley Brook here. So the restored site was downstream of that wetland in this region here. It's old pasture land, and it was very, very actively eroding. Our control site is near a farm just upstream of that location. So just a few pictures here to show you what the site was looking like before we started the restoration and the monitoring. So this is near one of one of the pools on the sites that was going to be monitored and restored and monitored. You can see the bank over here on the left side of the screen is actively eroding. And just to give you the rate of which that erosion was occurring, I mean, you can see there's new grass there, so it's eroding quite quickly. But when we were planning this study in the fall of 2008, we came out and we assessed this site as a potential candidate site for restoration. This tree you can see here, was actually on the far bank. So the river was actually running where the stream is, and this was over on the bank over there. So quite active in terms of the erosion, because this picture was taken 6 months later in June. So you can see, we've lost almost 4 meters of stream bank there. So it was eroding very, very quickly, far greater than you would expect in a natural system. So this is just a shot slightly upstream. This is at one of our study pools, what we call Pool 1. You can see that the bank is slumping inwards, and you can see the pool down here. It's not very deep. It came in at about 35 centimeters beforehand at low flow, so it's not a very deep pool. This is another shot. It's just upstream of our riffle, our study riffle. One of our study riffles, riffle 2, was actually just downstream of this picture, so just lower to the left. But you can see how over-widened the stream is here. We should be looking at a design width of about 4¼ meters for this stream. So the width of the stream should be about 4¼ meters, and we're up here. I think, if I remember off the top of my head, I think it measures at something like at 11 meters or something like that. It was just over 10 meters anyway. It was over 10 meters anyways for the width of the stream there. So quite over-widened and a very, very active system for sure. So what was our design? How did we actually go about doing this? We basically used a before-after reference approach that we looked at the system before we did the restoration for a year, and then we looked at it after we did the restoration for 2 years. And this was done at that treatment or restored site, and also at the control or reference site as well. Within each site, we did sort of visual assessments on the whole system, but we looked at 2 riffles and 2 pools in particular. So we had a total of 4 pools in the whole system and 4 riffles that we sort of monitored more closely. And those were chosen to be representative of the sort of habitat types that we were seeing along the whole reach. So what did we do? One of the first things we did was just basically use all the standard measures for quantifying stream morphology. So we went through, we looked at temperature, pH, size of pools, depth of pools, riffle to run ratios, sinuosity, those sort of measures. We made all those standard measures at both those sites before and after. We then went through and quantified the fish community. We did this twice a year. We did it in June, just after the spring migrations, and again in August during the sort of the low flow periods of the summer. We did the assessments using the regression method. That is to say, we netted off a region of the stream, and we went through and sampled it multiple times and got sort of a declining return that we could use to back calculate and figure out what the fish population was. We used 2 different methods just to optimize catch efficiencies. So on the riffle runs, we used electrofishing, and we used seining in the pools, just to make sure that we're getting the most optimal catch efficiencies. In the first year, we also tried some real basic catch per unit effort stuff, just minnow traps, essentially leaving them out for set time frames. This was mainly just to see if we were missing anything with our other sampling. So these minnow traps were positioned elsewhere in the reaches. And after the first year, we realized that we weren't actually catching very much of anything, and nothing different or in any different proportions than we were with our other methods. So this was stopped after the first year. In addition to the fish community, we also quantified the invertebrate biodiversity. So we did this using a modified Hess sampler, 3-minute samples. We sampled each of the 2 study riffles, so and each riffle was sampled 3 times. So, and then we did that sampling twice a year. We did it in June and December to get the 2 closure periods for the system. Then, because we do want to connect back to the underlying geological processes, we did quantify some of the sediment transport. Okay. We did quantify the sediment transport. Now, we did this in actually sort of 3 different ways to quantify what was happening with the substrates. The first method we used was sediment traps. So we buried, we established 2 transects on our riffles, and we buried 3 sediment traps on each riffle, and we would actually capture sediment as it was moving downstream. So from this we could figure displacement rates by filling how fast the jars were filling. And then by removing that material, we could run a mean particle size and organic composition test to figure out what type of material was moving and what size. Now the sediment traps are very good for small sediments and substrates, but they're not going to get the larger material movements. So to assess that at these 2 transects that we established on the riffles, we also put in bricks of different sizes and known weights. So we actually had 3 different weights, 3 times each, so 9 bricks in total. That we had put out along the transects, and then we simply monitored them over the summer to see how much they moved over time. And then lastly, to quantify what was happening with the substrate, we used quadrat assessments along these transects as well. We did that once a year just to see what was happening with the relative proportions of sediment that we were seeing in the stream. In addition to these sort of quantification methods, we also did a few experimental manipulations. The first was we did a leaf loading experiment. So essentially all we did here is we put out a little trap that we could load up with various concentrations of leaf litter to see what effect that would have on the benthic invertebrate community. So we monitored the benthic invertebrate community within the trap and out before and after the loading, and we just saw if there was any response. The basic premise here is that if the benthic invertebrates are nutrient limited, that if we were to provide them with nutrients, that we should see an increase in biomass as a result of that loading. So we did that at several different levels, and so that's the leaf loading experiment. In addition to that, we also looked at some litter retention work as well, where we looked to see if there was any litter retention in restored versus unrestored areas, if there's any differences, if that had any effect. So just a few pictures of doing the work. So this is the crew taking I guess it's a lunch break. I don't know. They seem to take a number of breaks from time to time, but anyway, so this is my restoration crew and our DFO supervisor of the day. We're just taking a bit of a break at that first site. Now, as my supervisor says, this is just another example of governance propping up failing banks. So you can groan at that one to your own pleasure. This is just an example of the fish assessments. So this is at our most downstream riffle. So you can see we netted off the area, and then we used electrofishing several times to get a drop in the number of fish, and then from there we can estimate the fish population. This is just an example of the guys doing the work. So you can see now this is after that eroding bank has been rocked. Now it's not just been rocked, it actually was designed to break up the downstream flow or the erosive force that was occurring and redirect it downwards to dig pools. So and then we put in some kickers and boulder groupings for cover as well. So so this is done, it was carefully designed by habitat biologists before as we did this. And then you can see the crew here putting in a digger log as well to help try and hold up this riffle and build that pool down below. And then this is just out on the reference site, once again, doing the fish assessments. So what did we find? The results, basically, if we go through in terms of our standard measures at the reference site, there was no changes. We didn't have any changes in pool size, distribution, no real significant changes anywhere throughout the reference site. At the restored site, O2 and pH remained stable. They were always sufficient values, so there was no times of real concern there. So, but there was no changes either. We did see there was creation of 2 new pools, so 2 pools that were not in that reach beforehand were established. One of which was established right at the top end of one of my electrofishing sites, and we actually ended up having to move the electrofishing site downstream a few meters so that we were still in proper riffle habitat and not in pool, because the pool actually encroached on that site. Another existing pool, that pool 1, actually doubled in its length, and 5 of the extant pools increased by their depth by at least 25 centimeters. So as I alluded to earlier, the pool, at least that pool 1 I said was at 35. Basically the pools ranged at 30 to about 40 centimeters deep during low flow period, and afterwards they were consistently coming in during low flow at around 70 centimeters. Some were even more substantial. One in particular, one of our study pools, it actually deepened to over a meter deep during the low flow period. So it's actually a really nice pool. In addition, we saw stabilizations of several gravel bars. One in particular, it was coming in it was consistently at a wetted width of about 8 meters. And then in the last year, it was down to about 4 meters wetted width. We saw the establishment of herbaceous vegetation, which hadn't been noted on that particular gravel bar before. And this was right next to one of our study sites. So that's why we mentioned this one in particular. But certainly we were seeing some improvements to habitat under a lot of our standard measures. And lastly, as we were doing our depth profiles as part of our standard measures, we saw that the thaw white position seemed to be quite stable in response to the so this is just the temperature data for the 6 different, the 2 different reaches, the restored and the controlled, for the 3 different years. This is mainly just to give you an impression of sort of what was happening. So you can see in some years we definitely were getting quite warm, and other years we were quite cool. When we plot out a sort of an average of the mean daily temperature, we see that a couple things. First off, that the restored sites here in the lighter colored bars were consistently warmer than the reference site, both before restoration and even after restoration, and that there was significant difference between years. So as the years progressed, we actually were getting cooler. 2009 was a particularly warm year, and then we had cooler summers in 2010 and 2011. Of interesting note here, though, is that if we look at the slope of these lines, we see that the rate of cooling on the restored site was actually greater than the rate of cooling. So there's a significant difference between restored and reference with respect to the years. So that certainly is a nice positive sign. Now, we did have a few confounding variables. The most notable one was occurred in that marsh. So as I set up where the study site was, I showed you where that marsh was, where the highway was coming in. We were doing an assessment on that marsh as part of another study, and what we found was that there was a fairly substantial amount of suspended sediment being produced. When we calculated the rate of that suspended sediment, and you can see that here, there was definite plumes of it. When we calculated that out, it was 60 metric tons of suspended sediment per year was the rate at which that was being produced. So, and unfortunately, of course, that is between our reference and than our restored site. So that was coming into our system just upstream from where we were. So that's a bit of a, certainly a bit of a confounding variable that would have had some sort of an effect. If we look at our fish densities, we found lots of significant differences. There was differences between species at the different reaches, at the restored and reference reach, before and after restoration, and between species at the reaches before and after restoration. So those were all significant differences. If we want to sort of look at a summary of it, what we see is that Atlantic salmon density increased after restoration, both at the restored and reference sites, and overall. So we did see increases there, which was certainly viewed as a positive sign. But the increase was greater at the reference site than it was at the restored. So I think I said these are all expressed as fish densities per 100 square meters. So we definitely saw a greater increase. In terms of the brook trout, and that's what I mean by brook trout, there was no brown trout in this particular, we never caught any brown trout in this system. The brook trout that we captured, we saw the density almost halved after restoration at the restored site, but it doubled at the reference site after restoration. So that's sort of an interesting sort of result as well. We did see, in terms of the minnow community, and I've just sort of lumped here the sticklebacks, the dace, and the green chops all in together. We did see a major decrease after restoration at the restored site. And we were much higher at the restored site than we were at the reference site. And we believe that this is just a factor of the height above tide. The closer you are to the tide, of course, the greater number of minnows you would expect to find, greater number of suckers. So in terms of the absolute difference, in terms of numbers, that wasn't particularly concerning, but we did see this large drop in the minnow community or minnow densities. Now, as you're going to see here in a second, we did break this out into usage in terms of the riffles and the pools, and we definitely could see where that difference comes from, different habitat usage in the pools and riffles. So we'll talk a little bit more about that major drop in minnows here in a second. And suckers, there was no effect on suckers, whatever. We took measurements on all the fish as well, and that size analysis is still ongoing. I'm still doing that. So in addition to this, the absolute numbers, will actually also be able to comment on the what's happening with the relative distribution of different species. So the interesting effects that we did get is when we plot the density of non-salmonids against salmonids, salmon and trout, we come up with an interesting density relationship curve. So certainly at lower salmonid densities, the density of non-salmonids was low, and then we sort of reach an optimum, which sort of spikes around approximately around 75 fish salmonids per 100 square meter, which seems to optimize the density of other fish. Beyond that, it sort of tailors off, and we tend to move back into a salmonid-dominated system. If we look at the riffles, essentially we saw increases across the board. That's really what was significant here, is that before and after restoration, at both the restored and reference, we saw increases in all categories of fish, all species of fish across the board. So riffle density certainly increased. We ran a plot there to see if it would also show a relationship, and it certainly does, but it is a much lower density for the optimum. So down around 25, 30 salmonids per 100 square meters seems to give us our optimum. In terms of our pool densities, what we see is that we got a variety of significant differences. Now, there was no significant difference before and after restoration, or between species before and after restoration. However, these were sort of, you would probably call these marginal. The p-values for before and after restoration was 0.11, and the p-value between species before and after restoration was 0.09. So certainly we're at marginal in terms of the significance, but we didn't find a significant difference for sure. We did get significant differences when we start looking at between species, between reaches, species at reaches, reaches before and after restoration. Those were all significant. In large part, these were driven, numbers were driven by this substantial drop in minnows after restoration. Now, when we went back and looked at the data, we see that this really high density that we get of 270 minnows per 100 square meters, we see that it was really driven by two catches in August of 2009 beforehand. At the restored site, we sort of hit a school of sticklebacks. We caught 512 sticklebacks in the pool and 130 sticklebacks in the pool. So, and those were very, very warm days. So certainly that had influence on this number and has driven this up to a fairly large density. If we were to take that out, not something we want to do for conclusions, just because we have so few before data samples. But if we take it out, then certainly the numbers become, that changes a lot of the significances and the overall densities and relationships. If we look just at the salmonids and trout, we see that that the density increased. Once again, the Atlantic salmon increased restoration at both reference, but once again it increased more substantially after restoration. Perhaps suggesting that the improvement is allowed for greater mobility of the species and they're able to move higher up the stream. In terms of the brook trout, we did see some decreases at restored and increases at reference as before, but here in the pool densities we did see a slight decline in the overall numbers of trout, so that was sort of something to be aware of as well. And once again, when we plot the sort of the salmonid density against non-salmonid density, we find that there does seem to be sort of an optimum salmonid density to achieve maximum biodiversity. In this case, around 125 salmonids per 100 square meters seems to be roughly our maximum. We're running a little short on time, so I'll just sort of go things. The results for the invertebrates, that analysis is continuing. Sediment transport, using the quadrat methods, we did see the emergence of some boulder near the end of the study, so there was a slight shift, decrease in the sand content and a slight increase in the boulder content after restoration. In terms of the sediment traps, before restoration, the sediment traps were filling twice as quickly as the reference site, and then that disappeared, and they were at the same rate after restoration, so that was certainly a good sign. And the large substrate movements, we were seeing more large substrate movements. This was using those bricks as an analysis, but we're still working on how to best present that analysis so that generally I can say the trend was to see a decrease in large substrate movement, but that was still ongoing. And of course, the detailed sediment analysis on the particle size distribution and organic content, that analysis is actually also still ongoing. Basically, just because we're running short on time, I won't talk about what we're getting on this one, the preliminary analysis, but it's more or less ongoing, the leaf litter loading experiments. But you can see here the traps that I was alluding to, and so these would have been loaded at different densities, and then we sampled in the traps and outside the traps over time. Some of the traps were remained open as well to allow fish access, so we could look at the presence of fish and the effect that had on the bird community as well. So in general, what are my conclusions? Well, in terms of habitat, we successfully saw creation of new habitat using our standard measures. Under those definitions, we got improvements to extant habitat, and we had new pools created. So we definitely saw creation of salmonid-based habitat. In terms of the fish utilization, we can positively say that we saw changes in distribution of fish within the stream. In generally, the salmonids increased, the minnows decreased, and the suckers remained stable. The overall population of trout was more or less stable as well. So that's generally what we can say were our general trends and conclusions there. Very interestingly, though, we found that relationship between salmonid and non-salmonid density, and so there seems to be some sort of optimum for biodiversity, and that has very important implications for restoration management and strategy development. That we want to make sure that we are, if our aim or our goal of restoration is to maximize biodiversity, as many groups say that it is, then we should be actually targeting a specific salmonid density. Unfortunately, we can't make a lot of really strong conclusions about the fish utilization because of our confounding variables and some of the trends. But one of the things that we believe is that if we have a longer data set, that we would get, we would certainly see that would help us make that conclusion better. So that's one of the conclusions we have here, is that this work really does need to be continued so that we can actually get a more stable trend. And then lastly, we can conclude that we saw differences in habitat utilization. So there was definitely differences in terms of the species composition between pools and riffle habitats. And so there's definitely something worth exploring there as well. In terms of the sediment transport restoration, we did see increases in particle size as estimated by the quadrant method. We did see a reduction in material movement, coarse material movement, but there's still a lot of work to do before we can make a final analysis on that. And lastly, the litter retention and inverted biodiversity. That analysis is still ongoing. So where does that leave us here now? Well, we still basically I have to finish off the invertebrate and substrate analysis. We need a little bit more information on some of the specific habitat types and utilizations. More work needs to definitely, we think more work needs to be done on non-salmonids, because there certainly is a connection there with restoration, and that biodiversity optima needs to be investigated. As I was alluding to in my conclusions, we certainly need a longer data set in order to draw more definitive conclusions about what's happening with response to restoration. Now, we're fortunate that this has actually been begun. There's a 10-year study being done by the Nova Scotia Department of Fisheries and Aquaculture, Inland Fisheries Division, that they're looking at 5 years before restoration, 5 years after, in a similar sort of design. So we're going to get that long-term data set to help draw those conclusions, which is nice. And especially with the trout, we're wondering there with the changes that we were seeing, the shift from the restored up to the reference in terms of the densities, that perhaps we need more information on what's happening with trout movement patterns and their movement patterns in response. Now, once again, we're fortunate that there's a PhD candidate named Aaron Spars at Dalhousie that is working on trout movement patterns in the system as well. So hopefully we'll get a little bit more information on that as his work concludes. I'll end off with just a few thank yous. My funders: Nova Scotia Transportation Infrastructure Renewal, Habitat, St. FX, DFO, Nova Scotia Liquor Corporation Adopt Stream Program, and Atlantic Salmon Conservation Foundation all contributed to this project. So we thank them for their financial support. And then there's many people that I have to personally thank for their assistance in